This is a video walkthrough for the lesson titled Bacteria from the Key Stage 3 topic Unicellular Organisms. This is the third lesson of the topic. What you're going to need for today is a pen, pencil, some paper or a book, and you may have also printed off the worksheet which is attached to Frog. Okay, so if you could record the title Bacteria, and I'd like you to have a go at starter A and or starter B. So looking at the foods that are numbered 1 to 5, we've got 1 Marmite, 2 piece of steak, 3 some nuts, 4 some yoghurt and 5 cheese. I'd like you to divide the foods into 2 categories, those that are made using microbes and those that are not. Or starter B, I'd like you to think of any other foods, apart from the ones that you can see pictured above, that are made using microbes. So if you'd like to pause the video now and have a go at doing that, please. So today's session is obviously all to do with bacteria. And by the end of today's session, you should be able to describe the functions of the parts of a bacterial cell. You should be able to tell me how bacteria reproduce and explain why bacteria are used to make yoghurt and cheese. Okay, so let's have a look at your responses to the starters. So if you chose to complete starter A, you were asked to divide the foods into 2 categories, those that are made using microbes and those that are not. So those that are made using microbes are your yogurt and your cheese. And without microbes, you should have listed there the Marmite, steak and the peanuts. So yogurt and cheese are made using microbes because different types of bacteria are added to milk. The steak, the nuts and the Marmite are not made using microbes. Marmite's actually made from concentrated yeast extract, which is a byproduct from fermenting or brewing beer. This means that it's made from a microbe, yeast, rather than actually made by a microbe. So Marmite was a little bit of a tricky one. Okay, what about other foods that are made using microbes? So we already know that yogurt and cheese are made using microbes. Some of the other ones that you might have thought about, alcoholic drinks. So wine and beer are made by yeast fermenting the fruits or cereal in the case of beer. Bread, that might be a common one, and you may have even made bread. And hopefully you'll remember that you used yeast to make the bread, and the yeast perspires, it ferments and produces bubbles, and that's why bread has got that bubbly texture. Other fermented products that you may have thought of are sauerkraut, if you've ever had that before, olives, and there's a few other fermented foods that you could have added there. We'll look at the term fermentation in a little bit more detail as we go through the session. So, thinking about bacterial cells. Okay, so we're going to have a little bit of a think about the structure of bacterial. You can see lots of bacteria in the photograph. The cells are actually made up of a bacterium called E. coli or Escherichia coli. This bacterium is typically found in a number of environments, including food, soil, and inside animal intestines. You might have noticed that I've been using the terms bacterium and bacteria. Bacterium is singular and it's used to describe just one bacterial cell, whereas bacteria is plural and it's used to describe more than one bacterium. So the picture shows some bacteria. Bacterial cells are much simpler than animal and plant cells. They have cytoplasm, a cell membrane, a cell wall, and some genetic material. You might have noticed there that I didn't say nucleus. Now, what I'd like you to do is to use the information from this slide and what I've just said to you to draw what you think a bacterium looks like. So pause the video now and have a go at doing that, please. Okay, this is a generalised picture of a bacterial cell, and hopefully what you've drawn is fairly similar. If it's not, don't worry, I don't want you to change it or redraw your picture or anything just now. In fact, I'd rather you just put your pens and your pencils down, okay? So you'll notice that I have labelled some of the organelles with letters. We've got A, B and C. And those labels are also found in plant or animal cells, and I'd like you to have a think about what they could be. And in addition to that, I'd also like you to have a think about what the functions of those parts could be. Okay, so hopefully you've recognised A as being cytoplasm. Cytoplasm is the jelly substance that's found in plant, animal and bacterial cells, and it's where those chemical reactions take place. Okay, B is pointing to the outside of the bacterial cell, and that is the cell wall, and that provides support for the cell. Obviously, animal cells do not have a cell wall, but they do, along with plant and bacterial cells, have letter C, which is the cell membrane. And if you remember from the work that you did in year 7, the cell membrane controls what enters and leaves the cell. Okay. So there are also some additional organelles in the bacterial cell that you don't necessarily find in plant and animal cells. So we have a loop of DNA, so it's a circle of DNA which is known as plasmid DNA. We also have some chromosomal DNA, which is a bigger loop of DNA which contains most of the important genetic material for the bacterial cell. Now you'll notice that the DNA, whether it's chromosomal or whether it's plasmid, is not contained within a nucleus. So bacterial cells do not have a nucleus, but they do have those little hair-like structures which are known as flagella. Now these structures that look like hairs or whips basically allow the bacterial cell to move itself, so they allow mobility. Not all bacteria have them, but it is a general feature of some of them. Okay, so what I would like you to do now is to redraw your bacterial cell, and I'd like you to label all of the names of the organelles, which you can see are lined on the picture, and then I'd like you to underline the features which are not found in plant and animal cells. So those are the features that are kind of unique to bacterial cells. And then, if that's too easy for you, which I'm sure it will be for some of you, I'd like you to have a go at the extra challenge. So I'd like you to have a go at explaining how DNA found in human cells is different to the DNA that is found in bacterial cells. Okay, so pause the video now, please. Okay, so how did you get on? We'll go through the names of the organelles, and if you need to add anything to your diagram or change your diagram, please do so. Okay, so our cytoplasm, cell membrane, cell wall, the flagella, plasmid DNA and chromosomal DNA. And you will notice that the plasmid DNA, the flagella and the chromosomal DNA are both underlined. Okay, so they are found only in bacterial cells. Now, you may be kind of let off from thinking that flagella might be found on animal cells as well, because there is an exception to the rule. Flagella basically help a cell to move, to be mobile. And there is a cell that you will have learnt about in year 7 that does need to be mobile in humans, and that is a sperm cell. So a sperm cell has a tail or a flagellum, and that helps it to move it or propel it through the female reproductive system to find the egg. So if you haven't underlined your flagella, I'll let you off just this once. Okay, the extra challenge was to explain how the DNA found in human cells differs from DNA in bacterial cells, and that is to do with where it's stored and the form of it. So DNA in human cells is stored inside a nucleus, and it's in straight linear lines known as chromosomes. Whereas in the bacterial cell, the DNA is free in the cytoplasm, and it can be found in circles, or it can sometimes be in a straight linear form as well. Okay, if you need to pause the video now to add anything extra to your diagram, please do so. Okay, so you may have also noticed that I've been using the term flagella and flagellum. Okay, so flagella is the plural of flagellum. So you'll see that the bacterial cell on the left-hand side has many whip-like structures, so it has many flagella. But the ones on the right don't have any whip-like structures at all. So they don't always have to have a flagellum, singular. If you'd like to know a little bit more about flagella and how they work, I'm going to put the link for the video that you can see on the screen into the notes bar, and you can have a look at that either now if you wanted to, or later on after you've finished. So flagella is plural and flagellum is singular. Right, okay. So I would like us now to have a little bit of think about what we already know about plant and animal cells and compare these to bacterial cells. So you may have already printed this table off, but don't worry if you haven't, you can just quickly sketch it out into your book right now. And I'd like you to take each of the cells and put a tick or a cross next to the organelles to show whether they are present or absent. Okay, so pause the video now, please. Right, starting with the animal cell. Okay, so an animal cell does have a cell membrane, it has a nucleus, it doesn't have any plasmids because the DNA is bound up inside the nucleus. It can't photosynthesize, so it has no chloroplasts, no cell wall, but it does have cytoplasm. Okay, so thinking about the plant cells, yes to the cell membrane, it does have a nucleus, no plasmids, chloroplasts, yes, it does photosynthesize, it does have a cell wall, and it also has cytoplasm. So how are the bacterial cells comparing? Does have a cell membrane, no nucleus, because the DNA is free, remember, within the cytoplasm in a plasmid. Can't photosynthesize, there's no chloroplasts. It does have a cell wall and it also has some cytoplasm. Okay, please mark your answers. Okay, so here you can see a photograph that is taken using a light microscope, and there are some cells which have been magnified about 100 times. Okay. What kind of cell do you think it is? Could it be an animal cell? Could it be plant or bacterial? Which organelles are you looking to identify to help you decide which one of those cells it could be? I'm hoping that you can see it's quite a thick line there. And I'm also hoping that you can see some green organelles. So that double line suggests that maybe there's a cell wall and a cell membrane. So that might be making you rule out an animal cell. So that leaves plant and bacterial cells. Those green organelles, I'm hoping by now that you recognise those as being chloroplasts, which rules out a bacterial cell. So, yep, it's definitely a plant cell. Okay, so you have now been able to describe the parts and the function of a bacterial cell. We're going to go on now to have a look at how bacteria reproduce. So let's think about them in a little bit more detail. Okay, so you can see my bacteria here, okay, and you can see that it is much smaller than a human cell. Typically, bacterial cells are between about 0.5 and 5 micrometers. And you can see the symbol for micrometers there just underneath the bacterial cell. Now, if you think about that in terms of millimeters, okay, which you're probably more familiar with, that is about 0.0005 millimeters. So that's about the size of the smallest bacterium. Tiny. Whereas animal cells are slightly larger. Okay, they vary between about 10 and 30 micrometers. So that's about 0.1 to 0.3 millimeters. So slightly larger, but still tiny. Now, bacteria are so small that you can't see a single bacterium with the naked eye. You can only start to see bacteria when they start to grow in groups, and we call those groups colonies. And each bacterial colony that you can see on the screen is made up of millions of bacteria. Bacteria use a method of asexual reproduction, okay, and that method's called binary fission. In this process, one cell grows and splits evenly into two identical parts. Now we're going to watch a little animation that shows you this in a bit more detail. And as you watch that animation, I'd like you to think about the answers to the 3 questions that you can see in the lower half of the screen. So, if bacteria have enough nutrients in a suitable temperature, how often can they divide? What do bacteria need to grow? At what temperature are the microorganisms kept in labs and in school? Okay, so I want you to watch the next slide with the animation on, and then we'll come back to those questions. If there are enough nutrients and a suitable temperature, bacteria can reproduce very quickly. In fact, they can double in number every 20 minutes. 1 becomes 2, 2 become 4, 4 become 8, and so on. So within just 1 hour, one bacterium can become 8. This happens through binary fission. First, the bacterial cell elongates, and then the DNA is copied or replicates. plasmids move to each end of the cell, and a thin septum develops in the middle. This septum then pinches off and two bacterial cells are formed. They are identical, or clones. Bacteria can be grown in the lab using either a broth solution or using an agar gel plate. The agar jelly contains all the nutrients that bacteria need to reproduce, so carbohydrates, proteins, minerals and vitamins. An inoculating loop is used to transfer the bacteria to the agar jelly. And to ensure there's no contamination, the loop is first sterilised by passing it through a flame. The Petri dish has a lid attached which is taped over, and then it's stored upside down to cultivate the bacteria. They grow fastest at warm temperatures. In a school, they would be kept in a safe temperature of about 25 degrees Celsius. They would grow faster at 37 degrees Celsius. However, there would be a danger of cultivating harmful pathogens. Okay, so let's see how you got on. If bacteria have enough nutrients in a suitable temperature, how often can they divide? They can divide every 20 minutes. What do they need to grow? Carbohydrates, minerals, vitamins and proteins. And at what temperature are the microorganisms kept in labs and schools? They're kept at 25 degrees Celsius. So although the warmer they are, the faster they will grow, if they're kept at a temperature above this at school, it's very difficult to control the growth. And so in that case you could end up with bacteria growing that you didn't want to grow, which potentially could be harmful bacteria. Okay, now we know that bacteria reproduce asexually, and we know that they require carbohydrates, minerals, vitamins and proteins to be able to grow. The bacteria respire the carbohydrates, and they use a type of respiration called anaerobic respiration. Now, anaerobic respiration means without air. So they use purely glucose, and that is broken down to make a substance called lactic acid. And you'll notice there that there's no oxygen like we would normally expect with aerobic respiration. It's without oxygen, and the product is lactic acid. Now, this is a very useful product for us, and we'll see why. So we've had a look at the functions and the parts of a bacterial cell, and we've also had a look at how bacteria reproduce. I'm going to move on to our final objective now, which is to be able to explain why bacteria are used to make yogurt and cheese. Okay, so lactic acid bacteria are used to turn milk into yogurt. The bacteria that's put into the milk use a sugar in the milk called lactose, and they respire it anaerobically to produce lactic acid. And it's that lactic acid that turns the milk sour and makes it a little bit thick. Now that is the reason why yogurt, if it hasn't got anything else added to it, no other sugars added to it, tastes quite sour, okay, because of the lactic acid. And you might have heard of the term curds and whey before. So the yogurt is like the whey bit, and the curds that are produced, the thickened bit, are then taken and more bacteria is added, which makes it to thicken more, and that makes it into the cheese. Now there's a really good video that you can watch that explains this process in a little bit more detail, which again you can either stop and watch it now or watch it after the video, and I'll put that in the notes bar. Now you can see some lovely cheese at the top of the slide there, and you might be interested to know that the particular species of bacteria that's used to make that cheese is actually taken from in between somebody's toes, which just goes to show you any bacteria or many different species of bacteria can be used to make cheese. Now, thinking about what you've just heard, I'd like to have a go at answering those 3 questions, please. So pause the video now and answer those questions. Okay, so the respiration that occurs in the bacteria is anaerobic respiration. If you remember, that means respiration without using any oxygen. So the glucose is directly broken down into lactic acid. And this is an example of a fermentation reaction. So this produces lactic acid, okay, and if you remember from some of the work that you did in year 7 with acids and alkalis, acids taste quite sour, so lemon juice, for example, tastes quite sour, and that's the reason why our yoghurt tastes sour. Now, that lactic acid has 2 effects on milk. First of all, it makes it sour, and then secondly, it thickens the milk, and that's where the term curds and whey come in. And curds are the thickened parts of the milk. Okay, so we have run through all of our 3 objectives now, okay, and hopefully you are able to describe all the parts of the bacterial cell, tell me how those bacteria reproduce and why they're used to make yoghurt and cheese. The last thing that I wanted to leave you with was some Petri dishes, okay. These Petri dishes show groups or colonies of bacteria that have been grown, and they've been grown on agar jelly, but the agar jelly has been coloured by different food colourings or dyes. And I just wanted you, after we've been talking about bacteria between toes, I wanted you to realise that some bacteria can in fact be quite beautiful. See you again soon.